Have you ever wondered how scientists ensure food and feed safety in a microbiology lab? It's a fascinating world where the smallest organisms play a huge role. Ensuring food and feed safety involves a detailed dance with microbiology, where sterilization, media preparation, bacterial counting, and identification techniques take center stage. These activities are vital in identifying and controlling potential threats like food spoilage bacteria, food poisoning bacteria, and more. It's a complex process, but don't worry, we'll break it down. Now let's dive into the step-by-step process of how these activities are carried out. First, all consumables must be sterile to prevent contamination. This is achieved through various methods of sterilization, such as direct heating, dry sterilization, filter membrane, and autoclaving, which uses high-pressure steam to kill all forms of life and biological agents. Once everything is sterilized, we can proceed with the preparation of media and reagents. This process involves mixing nutrients, such as sugars, salts, and amino acids, in distilled water, and adjusting the pH for optimal bacterial growth. It's a bit like cooking a nutritious meal for bacteria. Now our lab is ready. Let's delve into the intriguing world of microbial enumeration. To count bacteria and fungi, we use a technique called serial dilution. This involves diluting a sample multiple times and then spreading these dilutions onto specific media. It's like taking a bacterial selfie and counting the faces. The incubation period that follows is not just a waiting game. Specific incubation times and temperatures are key to promoting the growth of particular organisms while inhibiting others. It's a bit like setting the stage for our microbial stars to shine. Our next steps involve the isolation and identification of food spoilage and food poisoning bacteria. This is done using cultural media, biochemical, and serological techniques. These techniques allow us to categorize bacteria based on their appearance, biochemical properties, and antigenic composition. It's like an intricate detective story, solving the mystery in the world of food microbiology. We also identify indicator bacteria and probiotics. Indicator bacteria tell us about the overall hygiene condition, while probiotics are beneficial bacteria that promote health. It's like distinguishing the heroes from the villains in our microbial saga. Our journey doesn't end there. We also perform microscopic identification of molds, an antibiotic sensitivity test, and a minimum inhibitory concentration test. These tests help us understand the microbial world better, equipping us to fight against harmful microbes and utilize beneficial ones. Each step is meticulously carried out to ensure the highest level of food and feed safety. It's a fascinating journey through a world invisible to the naked eye, but of immense significance to our health and well-being. Let's recap the journey we've taken inside a food and feed microbiology lab. We've explored various methods of sterilization, media and reagent preparation, and counting bacteria and fungi using serial dilutions. We've delved into the isolation and identification of food spoilage, food poisoning, and indicator bacteria, not forgetting probiotics. We've peered through microscopes to identify molds and conducted antibiotic sensitivity to determine the relevant antibiotic to be used against specific pathogen and minimum inhibitory concentration tests to determine the exact concentration of any substance that can be used to destroy unwanted microorganisms. All these activities play a crucial role in ensuring food and feed safety. Remember, every bite you take is the result of rigorous testing and meticulous work done in a microbiology lab. Food and feed safety, it's a target that we all must reach.